Here we are. Hi, everyone. Welcome to RBCM at Home. My name is Kim Goff, and I'm a learning program developer at the Royal BC Museum. I'm coming to you today from my home, which is located on the traditional territories of the Songhees and the Esquimalt First Nations, the Lekwungen-speaking people in Victoria, British Columbia. RBCM at Home started in March when our museum and archive closed due to the pandemic. It was an opportunity to talk to staff about what they were working on from home. Now that some of us are back at the museum, it continues as an opportunity to talk to folks from the museum and to reach out to those of you at home or at school around BC. Today I have two guests. The first will be familiar to those of you who tune in regularly. Dr. Victoria Arbour is the curator of paleontology at the Royal BC Museum. And our second guest today is her intern, Alexis Bazinet, a recent UVic graduate and NSERC USRA student working on fossil feather anatomy in the collections at the Royal BC Museum. But before we hear more about the fossil feather anatomy project that Alexis is working on, Victoria, can you get us all on the same page about the relationship between birds and dinosaurs?
Yeah, absolutely. So I am a dinosaur researcher. And so you might be wondering, like, oh, why are we looking at bird feathers? And the secret trick is that all birds today are living dinosaurs. So when we look at the evolutionary relationships of things like chickens or ducks or eagles, all of them can trace their family lineage right back to meat-eating dinosaurs, closely related to things like Velociraptor. And then if you go even further back, it includes animals like T-Rex. So basically take like a T-Rex or a Velociraptor and make them extra, extra feathery and then very small, and you get a bird. So we're still working on dinosaurs for this project, just not the big Mesozoic type that we would normally think of. I hope whenever you look out your window and see birds in your backyard, you will think of them as little dinosaurs.
Well, I think I first heard about this theory, again, back, was it the 90s, early 90s with Jurassic Park? Has the theory been around longer than that, or is it something that's still debated?
It's actually been an idea that's been around for even longer than Jurassic Park, probably close to 100 years. People notice the differences in the anatomy, or like the similarities in the anatomy between some dinosaurs and today's birds. And then it really became much more prominent actually starting in sort of the 60s and 70s as an idea that probably a lot of dinosaurs were warm-blooded and some of them might have been fluffy, and they had very close relationships with birds, so much so that birds actually just descended from them directly. And then I think it really solidified in the late 90s when we started to find examples in China, especially, of things that were very clearly dinosaurs but had feathers. And they had either just sort of fluffy fuzz or full-on like flight feathers on their arms. And now it's basically like undisputed that birds are basically just a very, very specialized group of meat-eating dinosaurs.
Great. Well, thank you. I'm glad we're all on the same page in that regard. Yeah, if folks have questions, please use your chat functions and we'll pass those along. But next we're going to jump to Alexis. And Alexis, welcome. Thank you for joining us. I think you said you're joining us from Prince George today.
Yeah, so originally when I connected with Victoria about the research project, we were going to be at the museum, kind of behind the scenes, helping to catalog the new collection. But because of COVID, we've transferred our project to remote learning. So I'm actually here in my hometown of Prince George.
Well, welcome. And tell us what attracted you to this internship.
Okay. So I in my final year at UVic, like you mentioned, I had applied for an NSERC USRA, which is an undergraduate research award. And I saw Victoria had posted a project, and I did my minor in the School of Earth and Ocean Sciences, and paleontology was just always sort of out of my grasp at the school. And I was like, oh, this would be so much fun to work behind the scenes at the museum and to learn something new. And yeah.
Great. And I believe you've prepared a bit of a presentation for us because, as you're saying, you haven't been able to come into the museum. You've been working virtually. So I think you have some great images and, and things to share with us here.
Yeah, we can get started. I'll just share my screen. Okay, how's that? Good, okay. I'll just introduce myself again. So my name is Alexis, and I have been spending the summer working with Victoria Arbour at the Royal BC Museum, looking at bird and feather fossils. I'm just moving our little photos. So I'll just introduce you to our study site. So the McAbee fossil beds are located about 13 kilometers from Cache Creek, BC. We can see on the little map on the bottom right, off Highway 97. And it actually was declared a heritage site by the BC government for recreation, education, and research purposes. So lucky us. And the Royal BC Museum received a donation of about 18,000 fossils, which is a lot, from the Leahy and Langdon families who were previously collectors. And the McAbee fossil beds are actually part of the Okanagan highlands region, which is this region of a system of ancient lake beds that extends from British Columbia down into the United States. And as a part of our donation, we received many plants, insects, fish, birds, species new to science, and even feathers. So just a little bit more. So about our story kind of takes place 52 million years ago. So after the age of dinosaurs, studying the plants and insects in the collection, scientists were able to determine that it would have been a temperate climate at that time. So we can kind of picture the coniferous and deciduous forests of North America around this ancient lake today, instead of the dry cliffs we see. And I just want to give a shout out and a thank you to all the volunteers and staff at the museum and all the work that they have done on the Leahy-Langdon collection. So the last of the fossils was actually just unpacked at the museum in February of 2020. And without that, we wouldn't have been able to start our research project. So like I said, the Royal BC Museum received a donation from the John Leahy and David Langdon families in 2017. And staff packed up and transported the fossils to the Royal BC Museum, where volunteers unpacked, sorted, identified, and even painted little catalog numbers on all the fossils and placed them into these foam-lined cabinet drawers here on the bottom right. And this kind of leads me to our motivation for our study. So why are we looking at feathers? Well, feathers rarely fossilize in the fossil record because of their delicate nature, their little structures. But we actually have about 150 individual fossil feathers in the Leahy-Langdon collection, which is a huge sample size for fossils. And especially when you consider we only found about 2 partial skeletons of birds from the McAbee fossil beds. And we hope, Victoria and I, by measuring and studying these fossil feathers to provide more information about the biodiversity of ancient or extinct birds at the McAbee site 52 million years ago. So before I get to all our research projects, I'm just going to give a little brief background on feathers. So as we might suspect, feathers play a whole bunch of different functions we can guess at. They're for flight, display, so we all think about the peacocks down at Beacon Hill if you're from Victoria. Insulation, so keeping the bird warm. Weatherproofing, keeping the bird dry. And camouflage, and probably others that scientists haven't discovered yet. And depending on the body region the feather came from, there's different types, and I'll just run through how we can kind of pick those out based on the look and the shape from left to right. I don't know, maybe next time you go on a walk in the park and you find a feather, you'll know what it is. So we can have flight or wing feathers, or a fancy name might be ramages. And we can tell that these are flight feathers because they're asymmetric. So this leading edge, hopefully, can you see my cursor? Okay, is a bit shorter than on the opposite side. And one of the functions is to prevent the feathers from twisting in the air during flight. We can also have downy feathers, so these are loose, sort of short, and they trap air up close to the body to keep the bird warm. We can also have tail feathers or rectrices. So this interlocking fan shape helps the bird steer during flight. We can also have contour feathers. So these are the most common type of feather on a bird. They're body feathers, and they help keep it streamlined. So we can have, here's my mouse, this waterproof tip that kind of protects from the elements, and this more fluffy structure at the base. We can also have semi-plumes, which are fluffy for insulation and are sort of hidden under the contour feathers. Filial plumes, which help sense where the contour feathers are on the bird's body, and bristles, which I learned helped protect the bird's eyes and face. Okay. So we find all these different types of feathers in our collection, actually. So we can have flight feathers, a tail or contour. It's a little bit hard to tell which one until we start measuring. And downy, which are usually pretty easy to tell just because of how loose and fluffy they look. So we have all these different feather types. And feathers usually can have two sections. They can have a pennaceous section or a plumulaceous section. So every feather has a calamus that extends into a central rachis, or this long shaft that we can kind of think of as the quill of the feather. And off of the rachis, we have these branches called barbs. And off of the barbs, we have even more branches called barbules. And when we're looking at a pennaceous feather, like a tail feather or a flight feather, which is mostly stiff, flat, and has these hooks on the barbules that interlock and make this wind and waterproof barrier to help the birds. The rhyme I learned was fly and stay dry. Also, we can have plumulaceous feathers like the downy feathers, which have flexible, long barbules that trap air close to the bird's body for insulation. So when we look at individual feathers using modern microscope techniques, it can be used in a number of different areas of study. So forensics, aircraft collisions, food contamination cases, sometimes as anthropological artifacts, and even in prey remains analysis. So that's just a fancy way of looking at the diet of animals. And when we look at the barbules, which are, I would say, the branches of the branches, they have these little nodes and hooks that can form patterns and tell us a little bit about different bird families. But when we look at fossils that are 2D in the rock, we cannot always see all these fine details. So we have to rely on other measurements. So this is one, from McAbee, we had 2 partial skeletons discovered, and all represent bird fauna that were previously unknown in North America during the Eocene. So for example, we have a wing of a gaviform bird on the right, and this is evidence of a diving bird in North America at that time, which is pretty rare. And looking at the different skeletal remains from the Okanagan highlands in BC, at two sites, McAbee and Driftwood Canyon, we can get a better idea of what kinds of birds are around. So they found, like I mentioned, wings from McAbee of gaviformes, which is most closely related to the loon on the top left there. A postcranial skeleton from Driftwood Canyon of Songzadae, which is mostly closely related to rails on the top right. And two skeletons, one from Driftwood Canyon and one from McAbee, from coliformes and Zygodactylidae. So coliformes is on the bottom right there. And what's interesting is that there's about 17 extinct species with this widespread range in North America, Europe, and Africa during the Eocene, but there's only 66 living species alive today in Africa, found in Africa. And when we look at Zygodactylidae, they're sister to crown passerines. So on the bottom left, we have some photos of songbirds, and these were found in North America and Europe at that time. So looking at the Zygodactylid skeleton, the authors have labeled the bones with labels in black, and then Victoria and I went over and drew these red circles over where the feathers are. So 009 is this feather here, and I've just blown it up so you can see kind of what the preservation quality looks like. But the feathers are preserved in this halo-like fashion around the skeleton. And one interesting thing that the authors noted was the sulfurous coloration on the bones, which might hint at sulfide oxidizing bacteria. And these tend to occur on anoxic conditions. So the lake, that suggests the lake would have been oxygen free at the bottom, which might have had better preservation quality. And I'll just warn, so I'm going to talk a little bit about decomposition and fossilization. And so I'm going to show some bird carcasses, which might be a little bit gross in my next slide. So scientists conducted this experiment where they kind of tossed these bird carcasses to a bottom of a lake or freshwater body to help interpret what happens to birds during decomposition, and to help us interpret the fossilization process. And I'll just run through it really quick. So what happens to a bird after it dies, right? So this is kind of relevant to our story, because if we think of McAbee as this ancient lake, then you know, leaves would have fallen in from the trees, feathers from the birds, maybe, you know, some of the fish and insects would have died and sank to the bottom of the lake and got buried, and then over millions of years compacted and fossilized. But kind of what happens to a bird after it dies? So just run through really quick. So the carcass actually can remain intact for about 1 to 3 days. So it'll float up on the surface until bacteria starts to break it down, or scavengers start to get at it. So that's A on the top left. And then it remains complete, but as the soft tissues start to decay, we start to lose loose structures like feathers, like downy or contour feathers. And the skeleton may start to disarticulate. So that just means it starts to fall apart. So if there's little fish swimming in the lake picking at it, it'll fall apart. And so in C and D, we can see the skull has fallen off. And in E and F, which is a little bit more funny, the legs have come off. So we just have a picture of legs on the bottom right in F. And the last step is so the skeleton disarticulates, and then depending on the environment, if there's current, if there's scavengers, if there's lots of burial, the skeleton can actually remain mostly intact in the same position in the sediment, which is really interesting. So here I have, from the same study, we're looking at what happens to a bird, the muscle of a bird after 1 day of decay. So these are electron microscope photos. So Definitely not something you could see with the naked eye. So this is the muscle on A on the left. And in B, we have even more zoomed in, and we can see bacteria have already started to colonize the muscle. They're these little pill-like structures. And what I learned is that bacteria is actually already present in the gastric, circulatory, and respiratory systems of birds. And after they die, it starts to colonize from inside, and if the bird had any open wounds from the environment. So one interesting thing the authors noted from the study is that there was a bias towards these inland water habitats for preservation, and that wading and terrestrial birds were a lot more likely to be fossilized than true water birds, which is interesting because that sort of fits with what we've been finding so far at McAbee. But this can lead to differences in preservation quality. So not every feather is perfect. We can see the one on the top left has a fracture in it. The one on the top right, is sort of folded over or faded. On the bottom left, you can get frayed or missing parts, or sometimes the bottom right, is all dark and blurry. So sometimes there's feathers that you're not able to use, but definitely still a lot out of 150. So this is me working from home on the left, my setup, and this is Victoria's setup at the museum on the right. So what we have been doing is taking these high resolution scans under the microscope. So that's the microscope in our photo of the feather fossils. And we have been uploading them online so I can measure them here in Prince George. And I'll just show you quick. So we can see here the scale of the actual fossil in real life, and then the fine detail we are able to see kind of in the background once it's uploaded on the computer. And so we can take these photos and measure all the feathers in different regions along the top, middle, and bottom, and measure the, if you remember, I mentioned the barbs. So here again, we have a picture of a feather fossil, and we have the rachis. And then these are the barbs. So these lines are the barbs that branch off, and these other little lines are the barbules that branch off the barbs. And we can measure the barb spacing, which would be 2D in this photo, the barb diameter, which would be 2R, also the density and total length of the feather. And these measurements can tell us a bit about the bird. So they hint at differences in waterproofing and insulation in the feathers. And that can tell us a little bit about different bird families and correlate to things like behavior and habitat. So we measured many, many feathers. So here's a feather overview. And here's kind of what the measurements look like. So ImageJ is this free computer software where you can kind of upload a photo, set it to scale, and then drag a cursor and measure right on the photo. So you can see where I've measured here. One would be barb density, so sort of counting along the feather and seeing how many barbs branch off. The longer measurements are the diameter, or sorry, the spacing, and the little tiny measurements are the diameter of the barb. And we can actually see on the top left, we have barbs and actual barbules that we're able to see in some of the feathers. So you can see, actually, we do have some of that fine detail. But so we can measure these and compare to modern bird families and see if we can ID any associations with habitat or the species that were present. And what I have here is the only graph I'll show, but it's a box plot of the sum. So this little box is the summary of all the feathers, the many, many feathers we measured, and sort of shows this line is the average, sorry, and just like the spread of our measurements. And through this, we can identify patterns from all those numbers. And our results were a little bit unexpected, and we're still trying to interpret them. But what was interesting is we didn't identify bird families like we thought we would. We thought we would find, a close match, because we found, something close to a loon and that in the skeletons. But what we were really seeing is that there's differences, there appears to be differences between the feather fossils at McAbee and modern birds. So what I have labeled here on the bottom, the A, R, and T, are just birds categorized by habitat. So aquatic birds, riparian birds would just be things living around the lake, and terrestrial birds, so land birds. And what we see here, for example, is the barb density is a lot higher, which we don't know why. Maybe their feathers were different. And we also see that maybe their habitat was close to a riparian habitat, but still something's different. So we're still trying to interpret our results, but what we found so far is that the feather structure seems to, when you measure it, depends what region you measure, so it can change. Also, the feathers at McAbee suggest the birds occupied this aquatic riparian habitat, which kind of fits with the whole lake environment. We probably had wading and small perching birds, is what we're finding from all these little feathers. And that our data tends not to correlate well with the modern bird data. So this could be for a number of reasons. So there could be a lack of data to compare to, or there could be a taphonomic bias. So the study of taphonomy just means is the study of how organisms decay and end up in the fossil record. And so maybe there's some sort of bias, like in that decomposition study, how they mentioned bird and fossil feathers are usually found in inland water habitats, which is something that was unknown to me before. Or perhaps there is a true biological signal showing that ancient birds differed in their feathers compared to modern birds. But if you have further interest learning about the fossils, different kinds of fossils at McAbee, you can check out the museum's learning portal under fossil finds, or you can plan a visit to the Royal BC Museum. But thanks for listening, and I think we can take some questions if there are questions.
Thank you, Alexis.
See if I will just stop sharing my screen here.
That's perfect.
See all you lovely people.
So I'm not a scientist, but the first thing I wondered when you said there's a difference in the feathers, I wondered how much of that could be a result of temperature differences. Like we're in very different climates, and if one of the functions of feathers is about insulation, maybe there was so much warmer that could that be one of the factors that affects the differences you see?
It's sorry, I didn't mean to cut you off there, but no. It's a real riddle because we have a lot of tiny feathers and the barb density is also higher. So you'd think perhaps maybe it would be lower in this sort of like temperate forest, warmer environment. But McAbee is sort of unique. It's actually a little bit more upland than a lot of the other Eocene sites in North America at the time. So still a riddle, but could definitely be.
Alexis, what's the most interesting thing you've come across or surprising thing you've come across so far?
I think just for me was probably, so I did not come from a paleo background or a bird background, so it was a learning curve for me at the start, but probably all the different, just looking at the different feathers and all the different sizes, types, and the preservation quality, there's this real range that when you look through and you're just like, oh my goodness.
Yeah, and you said there's 100, I think, did you say 140 fossils that you have of
feathers? There's about 120 to 150 individual feathers preserved on these shale rock slabs. They're kind of squished in.
Right. Robert, who's watching here on Zoom, is asking how much of the differences have to do with evolution?
That's a good question. I was reading a little bit about, how Victoria was talking about at the start about dinosaurs and bird evolution. And I was reading an interesting quote the other day from a paper that was looking at dinosaur fossil feathers preserved in amber, so sort of like Jurassic Park. And it was talking about how, ancient feathers as they were developing, they had a lot of more different types and morphologies and little artifacts than modern feathers that have sort of solidified in their boxes. So perhaps maybe that is why our feathers are so different.
And Lori, also on Zoom, is wondering, Alexis, what type of research will you do next?
Oh, I don't know. I'm currently sort of looking into maybe looking at master's programs. So I originally did my degree in marine biology at UVic and took an interest in earth and ocean science, which kind of led me here. I feel like you always got to keep an open mind about what research opportunities are going to come your way next. And I have been personally always interested in marine mammals, but there's also people at the museum who study them. We study whale skeletons, so.
Mm-hmm. And here you are, as you say, doing something different, learning all about birds and feathers and getting this whole new area, and that's very commendable and remarkable. Victoria, a last question for you. I'm wondering, what are the success factors for this project? What will make this project a big red check mark in your books?
A big red check mark. That's a great question. Well, I should say, first of all, that I'm super impressed with the results Alexis has gotten. And I just want to also give a shout out that, Alexis has dived into this project without a background in bird anatomy or feathers, and also was very flexible about us shifting it to being completely remote. So Alexis and I basically got to have like coffee one day earlier in the summer when she was in town, and then everything else has been by Skype and Zoom and email. So I'm personally really happy with what we've done so far. And I think we actually already have that check mark on this project, because we have been able to get lots of measurements off of the fossils. And we didn't get the result we were expecting, but we got a surprising and interesting result. And that's actually a little bit more fun to chew on in some ways, because now we're getting to sit and talk about why our feathers are different in this way that we weren't expecting. So that's really, you have a research project with results that make you ask more questions is a good research project, in my opinion.
Absolutely. Excellent. And I think also this really demonstrates the value of internships and projects like these, and it allows such an immense volume of work to get done. It's such a big collection that you're working with. So congratulations to both of you for getting that internship rolling and for running it so successfully. I want to thank everyone for joining us. If you did join in late or you missed something and you'd like to go back, this is being recorded and you will find it on the Royal BC Museum's YouTube channel. So please have a look there. The museum has reopened and we are ready to welcome you back. Hopefully soon, Alexis, maybe you too can come. And you can find out more on our website about time, tickets, and exhibits. We will be continuing at home kids, and at outside for the foreseeable future. And links for all of these programs are on our website. Next week, we're going to be chatting with the curator of entomology and a student of entomology at the University of Berkeley. So we'll talk more about the relationship between research, universities, and museums. Until then, take care of yourselves and take care of one another. Bye-bye.